In our last few objectives, we were looking at proteins, and we talked about the sort of diversity of structures that you get in proteins. And one of the things that I mentioned briefly was that there's a special category of proteins called enzymes that have special functions in the cell. We're going to look at the enzymes now. We'll start by going back to where we were in Objective 32. So in Objective 32, we were talking about how there are many, many different shapes that allow proteins to carry out the different functions that they need to carry out. And we ended up with the shape of carboxypeptidase A and mentioned that it was an enzyme. So let's talk in some detail about what enzymes are and what they're capable of doing. Well, enzymes are proteins that are needed in all cells. For example, this shows you a map of all the complex metabolic pathways that are going on in all the cells of your body all the time. And if we focus just on the substances that are in the blue type here, then we see that they have names like pyruvate carboxylase or methylmalonyl-CoA carboxyl transferase or malate dehydrogenases, formate acetyl transferase. Notice all these end in ASE. It's not important that we know all these. In fact, that's why we have a biochemical pathways chart, is so that even biochemists don't have to learn all of these. They can just make reference to this chart when they need to look something up. But what we do need to know is what is going on with all these structures, these proteins, enzymes that are named here in blue type. Well, what they're doing is they're catalyzing chemical reactions. Back in Module 2, we talked about two different kinds of chemical reactions, those which require energy input, so we have to put energy into them to make them happen, and those that have an energy output, and we call those endergonic if we have to put energy into them, and exergonic if we get energy back out of them. So an example of a chemical reaction, and this is something that we mentioned very briefly in Module 2, but we're going to look at in some detail now, is the combination of hydrogen and oxygen, which makes water. So when hydrogen burns, it combines with oxygen in the atmosphere to make water, water vapor. We can see this process occurring rather violently in a famous video which I'll show you at this time.
Practically standing still now. They've dropped ropes out of the nose of the ship, and it's been taken a hold out down on the field by a number of men. The back motors of the ship are just holding it just enough to keep it. It bursts into flames. Wait, get it started. It's crashing terrible. Oh my, get out of the way, please. It's bursting into flames, and it's falling on the morning beds. All the folks would see that this is terrible. This is one of the worst catastrophes in the world. Oh, it's 500 feet into the sky. It's a terrific crash, ladies and gentlemen. The smoke and the flames now, and the flame is crashing to the ground, not quite to the mooring mast. All the humanity and all the fans just screaming around it. I don't even talk to people. Their friends are out there. I can't talk, ladies and gentlemen. Honest, it's just laying down massive smoking wreckage.
So let's break down what happened in this video that we looked at. There was a process of chemical reactions occurring. We said earlier that chemical reactions can either absorb energy or release energy. And you should know already which category these fit into. Whether they absorb or release energy, though, in each case, they have an activation energy. We can think of this as a little hump or a hill of energy that the molecules have to get over to get from the state where they are on the left-hand side, where they're reactants, where they're about to start a chemical reaction, to the state that they're on the right-hand side, where they're products. They've already gone through a chemical reaction. So that activation energy, as is shown here, is the energy that we need to absorb in order to begin the process of a reaction. Once that reaction process begins, then we form new bonds. Those bonds hold on to some of the energy, and we get products being formed. So the energy that's absorbed to start the reaction is whatever started this chemical reaction, and to this day we still don't know. It may have been a spark, it may have been sabotage, but it may have been static electricity. There have been speculation on all of those. But there was some kind of energy input that began the reaction. And then as those new bonds formed, as hydrogen molecules and oxygen molecules recombined in a different form to form water vapor, then we got energy being released as those new bonds formed. And it turned out the energy of the products was lower than the energy of the reactants. So energy was released. It's an exergonic reaction. As that energy is released, it comes back and interacts with the reactants on the left-hand side of this diagram. If we have enough energy to get those reactants over the activation energy, then they become products. Now we release more energy. We get enough energy out to cause those reactants to be able to get over that activation energy hump. And you can see that just as we looked at in examples in Module 2, that this is an example of a chain reaction. It's a reaction that doesn't stop until we finally get all of the energy out of the reaction, until all of the possible chemical bonds that can change have changed.
If oxygen and methane molecules are mixed together in the same container, they collide. But there is no reaction until a spark or flame is added. Why? To react, the molecules must collide with sufficient energy to break the oxygen-oxygen bonds and carbon-hydrogen bonds so new bonds can form. The additional energy needed to break existing bonds and start the reaction is called activation energy. An energy diagram can help show these energy changes. Initially, the oxygen and methane molecules collide but do not have sufficient energy to react. A flame can supply the activation energy needed to start the reaction. At this point, bonds break and new bonds form in an exothermic reaction that releases energy. The energy released supplies the activation energy for other molecules to react, and the mixture explodes. In the air, nitrogen and oxygen are continually colliding but lack sufficient energy to react. Heat from lightning supplies the activation energy to break bonds, and new bonds form in an endothermic reaction that absorbs energy.
Well, it turns out that it's a physical constant that we can't change. It's a property of bonds and the way that they're held together. How much energy is in the reactants, depending on what their individual chemical bonds are, and how much energy is in the products, depending on what their individual chemical bonds are. So we can't change whether a reaction is endergonic or exergonic. That has to do with the nature of the bonds between the different atoms in the molecule. And that's something we don't have access to. That's not something that we can change. That's not something that our bodies can change. That's just a physical constant. But what enzymes can change is the activation energy, is that hump that we looked at in the middle of this process. As we move through time from left to right, then they can change the size of that hump and make their reactions go faster than they would otherwise. In other words, they reduce the activation energy. In other words, they're catalysts. Catalysts, you may recall from chemistry class, are substances which reduce the activation energy, which make reactions go faster. Another view of this that may be a little bit easier to understand, if it's not, it doesn't matter how you understand it, you just have to understand it one of these ways that's presented, is the enzyme coming in and reducing the energy level that's needed for these blue balls to be able to get over this activation barrier. So what we need to imagine is these blue balls all bouncing around inside of this container, and that's the energy that they have. Recall that that's thermal energy. All molecules that are moving have a thermal energy. Sometimes that thermal energy is going to cause them to collide in such a way that a chemical reaction can take place. When that energy is sufficient to cause those reactants to come together in a way that reforms bonds, then they've gotten over this activation energy barrier. Enzymes are there to reduce the size of the barrier, and that's all that they can do. They can't change where the balls are in the left-hand bucket. They can't change where the balls are in the right-hand bucket. All they can do is change the size of this activation energy barrier. For that reason, we call enzymes catalysts. They bring together the reaction products by putting them in adjacent pockets of a protein which is folded in such a way that it holds these two reaction products together. And so we call that the active site, that little pocket that the reaction product fits into. The substance which is acted upon is called the substrate. So the substance that's undergoing a reaction is called a substrate. And if we add ASE onto the name of the substrate, then we get the name of the enzyme. For example, if we have an enzyme that breaks down the substrate acetylcholine, we'll learn about acetylcholine as a neurotransmitter later on in the course. If we have an enzyme that breaks down acetylcholine, the enzyme that does that is called acetylcholine esterase, because it breaks acetylcholine in an ester linkage. We've already learned what the ester functional group does. Some of those enzymes that we saw earlier then, we can make some sense of the names that we gave them. So in the case of pyruvate carboxylase, the substrate is pyruvate, and the functional group that it's acting on is a carboxyl group. In the case of malate dehydrogenase, the substrate that it's acting on is malate, and the functional group that it's acting on is a hydrogen ion. In the case of fumarate hydratase, or fumarase, the substrate that it's acting on is fumarate. In the case of citrate synthase, it's making something out of a citric acid molecule. So we had this picture on the previous slide, but we'll look at it in some detail. I like to think of enzymes as being the sort of matchmakers, or in Yiddish, the yentas, of the insides of cells. In step number 1, we have substrates or reactants that are binding to the enzyme's active site, and the active site then changes shape and brings those together. I think of this as like a male and a female in a relationship, two people that really, really should get together, and so the enzyme brings them together and invites them over for dinner at the enzyme's house at the same time, fits them in adjacent chairs at the dinner table. Then that allows them to interact with one another. If the interaction is favorable, in other words, if the energy that comes out of the reaction is greater than the energy that we put into the reaction, then the sites come into contact with one another. The product is released, and as the product is released, then the enzyme goes back into a shape where it can take in another set of reactants, and the process begins again. Now, enzymes are specific. They don't go putting together just any old pair of molecules together, or don't act on just any old molecule or pair of molecules, or sometimes 3 or more molecules. They have pockets that fit the shape of a particular kind of molecule or set of molecules that they're acting on. So if that molecule has any other shape besides the one that fits into the pocket, then the enzyme's not going to act on it. So this enzyme doesn't act on cylinders, and it doesn't act on cubes. It acts only on these complicated shapes that form the substrates of the reaction that this enzyme is going to carry out. In other words, the complicated shapes that fit into the pockets that this enzyme is designed for. Recall that the structure of the protein, because all enzymes are proteins, the structure of the protein is what's going to form this pocket, and the R groups of the amino acids inside that pocket are what's going to interact with the substrate and cause this catalysis to take place, cause this catalytic reaction to take place. So here's a concrete example from your Tortora textbook of an enzyme-catalyzed reaction. This is a kind of reaction that we've seen before. The substrates are sucrose and water. The enzyme which acts on the substrate ends in ASE. Remember, the substrate is the front part of the enzyme name, and it always ends in ASE. So this is an enzyme that acts on sucrose, so it's called sucrase. Recall that sucrose is a disaccharide made up of glucose and fructose. When the enzyme acts on this, then, it turns sucrose back into glucose and fructose. Now the glucose and fructose molecules are released. The reaction's complete, so the enzyme goes back to the shape that it was before and is able to take in another sucrose molecule. So in step 1, it has a shape that holds on to a sucrose molecule. In step 2, it rearranges slightly in order to make this reaction take place. In this case, the breaking apart of glucose and fructose. And in step 3, it changes shape back to where it was in step 1 so that it can now take on another sucrose molecule. This enzyme sucrase is actually the secret behind chocolate-covered cherries. If you like chocolate-covered cherries, you may have wondered how they get a liquid center into them. Well, the answer is that when they form them, the center is not liquid, the center is solid. The center is made up of sucrose molecules, which are solid at the temperature that the chocolates are made, but they've added in the enzyme sucrase along with the sucrose. And then as we store the chocolate-covered cherries over the next week or more, then the sucrase acts on sucrose, breaking it into glucose and fructose. Turns out that the glucose and fructose solution, unlike the sucrose solution, are a liquid at room temperature instead of a solid. So we go from a solid center to a liquid center.